This is 12.7 Antibody Notes. The essential question is, what are the 5 classes of antibodies and how do they function to provide humoral immune responses? Antibodies are also known as immunoglobulins, and it's abbreviated with a capital I and a lowercase g. They are soluble proteins and they are secreted by the plasma cells that derive from the B cells, which are involved in the humoral immune response. The antibodies are then carried in the body fluids such as blood. The antibodies are capable of binding to specific antigens and carrying out various types of reactions or effects in the immune response. The classic structure of the antibody is a Y-shaped structure that are made up of 4 amino acid chains, and they are linked by a disulfide bond. So these lines that you are seeing right here, those are your disulfide bonds. 2 of those chains are light chains, which means that they are smaller in size, so that's your light chain here. And then you have the heavy chain, which is the longer amino acid chain. That's your heavy chain. So the 2 heavy chains are then linked together, and what's holding them are the disulfide bonds. In the diagram, the V stands for variable region, which make up the top portions of the Y. Here is your variable, and here is your other variable region, and together they make up the antigen binding site. So this is the part where the antibody will attach to an antigen. The C in the diagram stands for constant. So those are variable. Obviously, it's called variable region because their shape in different antibodies are going to be different. Whereas the constant portion is going to be pretty much the same between all of the different antibodies or immunoglobulins. There are 5 classes of antibodies, and you can kind of remember the acronym GAMED to remember the 5 antibodies. And so they are slightly different in shape, and more likely they are different in their function and their role. There is IgM, which is the first immunoglobulin or antibody that the body produces, and it provides the primary immune response the first time you come in contact with a pathogen when you are pretty much a baby, an infant. IgA is found mainly in mucus and other bodily fluids, but mainly in the mucus. IgD is important in activation of the B cells. IgG can be the only one that crosses the placental barrier, which means that this is the example of what provides the example of the naturally acquired passive immunity, because the antibody is transferred from the mom to the baby. It's the only one that can cross the placental barrier. And then IgE is involved in allergies. Here is a more thorough breakdown of the antibody classes and the terms, the structure terms. Monomer, it means that you are looking at just one of those Y structure. Then when you have a dimer, so IgA is a dimer, which means it has 2 Y structures that are hooked together. IgA have 2 different ones. They have IgA1, which is a monomer, and then IgA2, which is a dimer. So they have 2 different classes of IgAs also. Then the IgM is a pentamer, which means that it is a 5 of those Y structures that are all linked together. The most abundant antibody or immunoglobulin is the IgG, and it is involved in primary and secondary immune response. Remember, this is the only one that can cross the placental barrier to the baby, mom to the baby. The IgM is the one that has the pentamer shape and are involved in the primary response. Then you have IgD, which is probably the least known immunoglobulin out of all of the 5. And it is important in maturation of B cells and in activating B cells. And then IgA is the dimer, the 2Y shape. This is the one that's found in body fluids, more specifically common in mucus. And then IgE, which are the most common thing that you need to know about IgE is that it stimulates the mast cells, which then produce histamine that causes inflammation and involved in allergic responses or allergies. There are various ways that antibodies can kill or inactivate or disable the pathogen, and depending on the different classes, it can act differently. So one way is called the complement fixation, and so there is a special relationship between the complement protein that are floating around in the fluids, and they attach to the protein, attach to the antigen, and that can cause an immune response. So one way that happens is that complement protein attaches to the membrane, which causes a hole in the membrane of the pathogen, and then water is rushing into the cell, such as a bacteria, and then that will cause that bacteria to swell up, and then it will burst and then cause a cell lysis, and then which will kill the bacteria or the cell of the pathogen. Opsonization is what the antibody does. It actually binds to the surface of the pathogen or the antigen on the pathogen, and it's like putting a tag on the pathogen to kind of alert other immune cells, such as a macrophage or of a phagocyte, to either destroy it or alert other immune cells to come in and destroy it. Neutralization works on specifically bacteria. The way bacteria can be harmful to us is that the waste product of the bacteria, they produce a chemical, and that chemical can be poisonous to us. And that's what we call a toxin, is a poison. And what the antibodies do is that it can take those chemical, the toxin, and completely surround it or bind to it, and then it kind of makes it so that it is no longer poisonous to us. And that could also work with viruses. So if a virus is going to attack our cells, it will completely surround the virus, and then so now the virus can't do what it wants to do, so it neutralizes it. Agglutination is what causes the antibodies to do is that antibody, like IgM, which is the pentamer, has 5 areas where things can bind to. So you have bunch of antigens binding or pathogen binding to that, and you're going to create a much bigger blob of stuff. Then it's going to be easier for the other immune cells to be able to recognize and destroy. Agglutination also is what happens when you receive the wrong blood type, and then the clumping or clotting happens, and that's what agglutination is the clotting, the clumping of the blood cells. Precipitation is very similar to agglutination, but in this in this example, it could involve any of the monomers too. So what happens is that the various antibodies, they all kind of join together and create a one huge clump. Okay, and so if you have liquid like blood or any kind of fluid, what happens is these pathogen will become one clump, and they will actually will settle out and become a solid, insoluble, a solid material and liquid. So a pathogen can be very tiny. So in a mucus and things like that, it could still be part of the fluid. But when you have a big mass of cells like that, then it's going to settle out of the liquid, and then it's more easily able to be seen. Also, when we're talking about blood, where liquid is flowing very well, when you have this solid mass of cells, then it's going to settle out of the solution, and then it's going to stay in that place, and it's not going to be able to move. So precipitation prevents spreading of the pathogen to other areas of the body. So here are some of the functions, various functions of the antibodies. So you have neutralization, which is going to take something that is poisonous and make it not poisonous. And agglutination is when you are creating a clot or a clump of cells. And precipitation, when you are taking a liquid and becoming solid so it can't spread to other areas. And all of that, these actions can cause the macrophages or other immune cells to be able to recognize that there is a pathogen there more easily. Or it can create an inflammatory response, and which will release histamine, which will cause inflammation and cause an immune response to occur also. A complement fixation can also cause an inflammation, and also it can cause, remember that opening the hole in the membrane of the pathogen, which can cause cell lysis. Antibodies are actually used for research and clinical testing and for diagnosis of diseases, and treating and detecting cancer. So antibodies are used to deliver cancer-fighting drugs to cancerous cells, and then the levels of antibodies can detect pregnancy and certain kind of diseases. The antibodies are used as treatment, such as hepatitis and rabies. 12.7 notes homework. Number 1, describe the structure of the antibody. Number 2, which is the most abundant immunoglobulin? Which immunoglobulin can cause the placenta, and which immunoglobulin is involved in allergies? Number 3, what are agglutination, neutralization, and precipitation?